So what are the functions of the nucleotide? First you can remember is that the nucleotides join to form a long chain, and that chain would be called as the nucleic acid or polynucleotides. And then this nucleic acid can be either DNA or it can be RNA, and nucleic acid is important as the genetic material. Then the second function of the nucleotide possible is the energy currency. We already learned today that ATP is the energy currency, and remember NADP, NADPH, NADP plus and NAD plus, these will join with hydrogen, or you can say the protons, to give energy containing or reducing power. This, see, once they pick up the hydrogen ion, they become reduced. Now they are strong reducing powers because they can pass on this H plus to some other molecule and themselves will become oxidized and will cause reduction of some other molecule. So these play important role as the assimilatory power. You'll be learning about this power in the case of respiration chapter and photosynthesis. So they act like energy carrier. Then you can remember that we have learned cyclic AMP. Cyclic AMP is the secondary messenger which starts reactions inside the cell. In the case when the primary messenger is not able to go across the plasma membrane, for example, maximum of the water soluble hormone and the fat soluble hormone. This concept will come in last chapter of the 11th class NCERT. Then next is phospholipid formation. Even phospholipid formation requires nucleotides, and then coenzymes and vitamins, they also require this is coenzyme, also requires the nucleotides. So nucleotides play important role in the formation of these many various things. Now starts your topic of nucleic acid. So nucleotides are microbiomolecule, but nucleic acid is macromolecule. So what is macromolecule and why do you call it macro? Because it's a linear arrangement of heteropolymer. Now just like proteins, please pay attention, there also I mentioned this point there. Heteropolymer are those polymers which are not homopolymer. Reason, they don't have repetition of same thing, they have repetitions but of different things. That's why it is called as heteropolymer. So what is repetition of different thing here? See, phosphate will remain same in every nucleic acid. It would be either ribose sugar or deoxy. You cannot mix both of them. So phosphate is common to both DNA and RNA. Ribose is found in RNA and deoxyribose is found in the DNA. So the only variable thing is the nitrogen base, because nitrogen bases are of 4 types: A, G, C and T in the case of DNA, A, G, C and U in the case of RNA. So these are the things which are going to be repeated in different sequence. So that's why it is heteropolymer, because a DNA can carry AATTGC, it can carry CCGGAA, any manner it may join, and this joining will cause formation of a linear heteropolymer. So the variable portions are the nitrogen bases, rest whole structure of the nucleic acid is going to be similar. Now next is, this is the linear heteropolymer. It is also called as the polynucleotide, and remember nucleic acid is made up of CHNOP. That means the carbon is there, hydrogen is there, nitrogen is there, oxygen is there and the phosphorus. Now nucleic acid for the first time was isolated because by this time technology had improved a lot in the biology lab or biochemistry lab. So first time the nucleic acid was extracted by Friedrich Mischer in 1868 and 69, somewhere in between, from the pus cell. So he was studying the pus cells and he took out the genetic material, what he called as nuclein, because it was from inside the nucleus. That's why he called it as the nuclein. Later Altman discovered that it is acidic in nature. Altman discovered in 1889 it is acidic in nature, so he modified the name into nucleic acid. So nucleic acid, remember, is present in every living organism. It can be eukaryote or it can be prokaryote or it can be virus. All of them carry the nucleic acid. And the nucleic acid is of only two types, either it is DNA or it is RNA. And please remember, DNA and RNA both are present in eukaryotes, both are present in bacteria or prokaryotes, but only one out of two will be present in the case of virus. So remember, nucleic acid, that means DNA, and RNA is common to all eukaryotic cells. That means both DNA and RNA are present in all eukaryotes. Even prokaryotes will have both DNA and RNA, but viruses are the only organisms which will have either DNA or RNA. They cannot have both inside them. Now let's start with the slight detail of what exactly is RNA. Why slight? Because details of this topic will come in genetics unit of your 12th class syllabus. Complete chapter is there on this concept. So now what is RNA? RNA full form is ribonucleic acid. It is single stranded. Remember, DNA will be double stranded except for some virus, otherwise DNA is always double stranded. So RNA is always single stranded, with one exception. Again, there are some virus which will have double stranded. You mention like SS means single strand, DS means double strand. So the RNA is generally single stranded except for some viruses which may have double strand. Double strand means two strands will be there. The RNA. Then RNA carries ribose sugar, which is a reactive sugar, because of second position has a hydroxyl group attached. RNA is reactive again because of uracil is present. Uracil is reactive in nature, and the nitrogen bases present in the RNA are uracil and cytosine. These are the pyrimidines, and adenine and guanine, these are the purines. And RNA is formed by transcription inside the nucleus from the DNA. Please remember, DNA is the